Hi everyone, I'm Fatema Zahra and I'm a postdoc at Reinstam's lab at Leiden University. And my talk today is about the three-dimensional organization of the Syn3A chromosome. So our lab works on chromosome organization and dynamics. And over the past few years, the field has come to realize that bacterial chromosomes and some archaeal chromosomes are hierarchically organized as follows. So first, the entire chromosome is divided into macrodomains. These are regions of the chromosome in which the chromatin interacts more frequently with itself than with chromatin that is present outside this region. When we zoom into these macrodomains, we see the hierarchical organization where these larger macrodomains are formed by the interaction of smaller subdomains. And the boundaries between these subdomains are generally maintained by highly transcribed genes. And when we zoom in even further, we find that the smallest level of organization in bacterial chromosomes, and of course some archaeal chromosomes, are operon level loops that are formed by nucleoid-associated proteins. Now, this is rather interesting because these architectural nucleoid-associated proteins are able to detect changes to the environment, and they respond to the stimuli by altering the structure of the chromatin, and in turn, this affects the genome processes, for example, transcription. This principle makes Syn3A, or the minimal chromosome, rather interesting, because like, for example, a model organism, Escherichia coli, that has about 200,000 nucleoid-associated proteins in itself, Syn3A has about 230. And like was mentioned in the introduction last week, this means that the Syn3A chromosome is reminiscent of those chromosomes at the very early stages of chromatin evolution. And that makes it interesting because we now wonder how this chromosome is organized. So what are the most basic principles of chromosome organization? To do that, we looked at the three-dimensional organization of the Syn3A chromosome, and we did that with chromosome conformation capture. So Syn3A cells in culture were first fixed with formaldehyde because this cross-links the DNA to protein and protein to proteins, and this captures a snapshot of the chromosome at a particular point in time. We then extracted the chromosome from the cells and treated it with a restriction enzyme that digested this entire chromosome into DNA protein complexes. And those DNA molecules, or those loci that are close to each other in three-dimensional space, remain close to each other in this DNA protein complex. We then perform an intramolecular ligation so that these molecules form hybrid ligation junctions. We purify the library, sequence it, and the information contained in this library is then assembled into a chromosome contact map. So this is basically the latest version of the Syn3A chromosome contact map that we have. And this map is a probability matrix where every pixel, or in this case, it's a very noisy map, but in principle, every pixel represents the relative interaction frequency between the corresponding loci on the axes. So just so we all gather how to read this, this point over here shows the relative interaction frequency between a locus at about 170 kilobases and another one at about 150. So now that we know how to read this map, what exactly is the map telling us? The first thing we see is this primary diagonal of high interaction frequency. This arises because sequences that are close to each other on the primary structure of the chromosome are definitely close to each other in three-dimensional space, and they show a high interaction and thus the presence of this primary diagonal. The next thing that we observe is the absence of a secondary diagonal. And to explain this, I will compare the Syn3A contact map with those of other bacteria. So other model organisms like Colobacter crescentus, Bacillus subtilis, and even Mycoplasma pneumonia have this fainter secondary diagonal that lies perpendicular to the primary diagonal. And this is a feature characteristic of circular chromosomes, where the two halves of the chromosome interact over the length of the cell. And this is referred to as interarm interactions. Other model organisms like Escherichia coli lack this secondary diagonal, and they do not show, and so in principle, they lack interarm interactions. And since Syn3A resembles this organization, we expect that there are no interarm interactions in Syn3A, or no detectable interarm interactions. So this map also shows us that the chromosome of Syn3A is organized into domains. Now, domains are regions of the chromatin where these regions have a higher frequency of self-interaction than with regions outside the chromatin segment. And this appears as squares of high interaction frequency along the main diagonal, and that is rather visible over here. This region also shows you a hierarchical organization because you see these denser squares that collect into a larger one. The fourth feature that we see is that we start seeing evidence of loops. And for that, I will zoom into this region here. Now, a loop is a region in which two loci are anchored by a nucleoid-associated protein, and the intervening DNA remains free. And this appears in a contact map as these spots of these regions of high interaction frequency that appear off, like away from the main diagonal. And this means that, I hope you can see the cursor, that these two corresponding regions here are anchored to each other by a nucleoid-associated protein, and the intervening DNA loops out. Now, for me, this loop is rather interesting because it may have been formed or may form as a consequence of co-regulation, since this loop actually encodes a tRNA that, for glutamine, and it encodes subunits A, B, and C of glutamyl tRNA amidotransferase. Right. So these features that we see in the Syn3A map, which is the presence of a primary diagonal, absence of a secondary diagonal, the domains and the loops, they are already present in these model organisms. And in these model organisms, we are beginning to understand how these structural features, what they represent in terms of genome transactions. So, for example, in Colobacter crescentus, these regions that form the boundaries between the domains are formed by long, highly transcribed genes that physically separate the flanking chromatin and prevent them from interacting. In the case of Escherichia coli, this is the TER macrodomain that is compacted by the binding of the MET-P protein to the MET-S sequences. And the compaction is rather important for timing cell division and for ensuring proper segregation of the chromosomes into the daughter cells. But in the case of Syn3A, we don't quite know what these structural features represent. And that is what we would like to investigate. So the first thing we would like to do is we want to correlate these chromosome contact maps with protein occupancy studies, for example, from ChIP-seq analysis, to identify what proteins contribute to the structure of the chromosome, if they contribute to the structure of the chromosome. We also want to correlate this to the transcriptional profiles, just to observe how the organization of the chromatin relates to the RNA, to the expression levels. The second thing we would then like to do is we want to know how these chromatin binding proteins actually affect genome interactions or transactions like transcription. So we want to up and down downregulate these chromatin proteins and see the effect on chromosome architecture and thus the effect on transcription. And finally, because this chromosome is reminiscent of chromosomes at the very early stages of evolution, we would obviously like to see how nucleate-associated proteins drove chromosome evolution. So we would like to express non-native, or ectopically express DNA binding proteins in these cells to see how the chromosome architecture changes. And with that, I would like to thank all the people that contributed to this project, and I'm glad to take your questions. Thank you.
That is terrific, Fatema. I understand the whole system much better than I did before. If you like, we can arrange to perhaps alter expression levels of specific proteins. We had talked about restoring the HUP-A to its conventional level of synthesis. Its lower level of synthesis, we think, was an accident of the minimization process, but we can think about rebuilding cells. We'd have to go through transplantation, but we can rebuild cells so that proteins that you designate might be expressed at higher or potentially lower levels. Things we could do.
All right. That would be nice.
Do you buy my hypothesis that the absence of the relative absence of HUP-A might be reminiscent of a primordial, world without a lot of, basically without chromatin, or without chromatin as we know it today?
Yes, I think the absence of, or the presence of such low levels of HU, are reminiscent of very archaic chromatin, I would say. I had actually we had discussed this with Remus earlier, and it also seems like overexpressing HUP-A, since HU is expected to constrain supercoils. We expect that the domains would become much more visible, and the chromosome itself would be more compacted, since supercoiling helps compaction of the chromosome. So we would definitely see changes in the chromosome organization, which would be more, would be sort of closer to the model organisms that we see.